So before I start today, I just want to hear from you a little bit. How is it going? Do you have any comments in general? We were supposed to give you the surveys yesterday, but apparently we haven't done. Today we will give for 2 days, right after my session. And because some of you are already, you're leaving today, I think. And one thing we might do is to start right away to the lab session after the lecture. And I will follow Ahmet's trend. Probably I will also make a shorter presentation so that you will have more time for the applications. And while you are working, you can go and grab your sandwich, eat it, and come back so that we will save time, if you agree with it. Those who would like to take a longer break, 2-hour break, this is up to you. But I think today time is very precious since most of you are on your way, as far as I can tell from here. So I learned yesterday that there have been some problems when you first arrived. I didn't know about that. Then you had to go all the way up to the hill at the dorm and then come back here and then go a second time. I'm so sorry. I didn't know about that. And it's good that at least now I learned about it.
Good exercise.
That is the good part of it. That's what I told the person who...
It reminds us of flowers because we come down the mountain every time.
So I thank you for having such a good spirit, but this is something we may want to correct in future years, just to make sure that you either stay in a more convenient place, or we can arrange a transportation, a shuttle between. So that might help. Or a zip line. Yeah, we can create. So that was something I was not actually expecting, but that happened. So in general, please do ask us questions. Tim, Ahmet, Anindita, you met with her yesterday, right? Anindita and myself will be here till you leave today. And so we are here to answer questions as much as we can before your departure. Please, hopefully, we will be interacting very efficiently, very productively today. So this is something I've already shown yesterday. And I was mentioning that we would be moving to druggability and sequence specificity, these issues. And already Ahmet made a nice presentation on druggability. I'm sure many of you look forward to its applications in the hands-on session right after my lecture. Now, the point I would like to make here is the following. In the same way as experimentalists, they do multiple experiments, to answer a question, we should also take advantage of multiple methods. So NAMD is a very powerful tool. There is no question about it. But it's always useful to complement it with other alternative approaches, including those based on sequence. And if, for example, a given interaction appears to be important, then we should expect that interaction to be evolutionarily conserved if it has a functional significance, right? So that's one good way of trying to understand whether this is a conserved site or a site that co-evolves with another one. So we look at, for that purpose, the approach is very simple. Actually, biologists, they usually do it quite well. This is probably the first best thing they do, to compare sequences, do multiple sequence alignments, And it turns out that physicists, we don't have to, And I found it very useful to complement our structure-based studies with sequence-based studies as well. So the idea is you now you have to generate a multiple sequence alignment. And again, we have methods to even retrieve already existing multiple sequence alignments for different families of proteins. And then by examining the sequence alignments, now each column refers to a sequence position, as you know. And then we try to understand whether a given sequence position is occupied by always the same type of amino acid or not. So for extracting information on conservation properties. So there are many methods, sophisticated methods, and I'm not going to go into that within this lecture. But the simplest you can think of is the classical one, is what's called a Shannon entropy, where you, for each position, you simply look at the probabilistic occurrence or the fraction of a given type of amino acid. And you have 20 different types of amino acids. In some cases, the deletion is also considered as a 21st type. So you just look at their probabilities and have this type of summation. Okay? So if in the limit, you can think of if everything is, any amino acid is possible there, your probability of X is going to be so the notation, let me say, X is amino acid type. I is the sequence index. So XI, PXI means the probability of observing an amino acid of type X at position I. Okay? So if there is no preference for any type of amino acid in a, what we expect PXI for all types of amino acids to be? 1 over 20. And if we take the deletion as an incidence, 1 over 21. Okay? If it is also equally probable. So that gives you like the highest value that could be achieved. Higher entropy means it doesn't obey any rule. Okay? Or you can go, you can have the opposite extreme where you're going to have practically the same type of amino acid everywhere, and log 1 is going to be 0. Okay? So that is the first thing. And the second property that we consider is how pairs of columns co-evolve. Okay? And in the literature, you can see many different nomenclature. They call it correlation mutation, correlated mutations analysis, or co-evolving residues, compensating mutations. And again, a classical measure which is being used in information theory is mutual information. Now, this definition will be self-evident, I hope. It means the probability of observing simultaneously the joint probability of having amino acid of type X at position I and type Y at position J. Okay? So now we consider all 400, let's say, different incidences. And for each column, we have a probability, joint probability of observing different pairs. And we would like to see how much this joint probability differs from the singlet probabilities, okay? And this departure will be considered as a signal. So if this number is higher, you have a higher signal, these residues co-evolve. Okay. So that is essentially what the basic approach. But in the literature, it has been observed that actually these are very simple metrics. We need better measures than that. One, for example, that has been introduced in 2008 and has been very widely used is the following. Now, if there is, in the multiple sequence alignments, there may be some biases. Not all amino acids are equally represented. There may be some what we call common ancestry effects. All the family members may certain sequence positions may share some properties, and we would like to get rid of them when we evaluate the coevolutionary patterns. So we want to correct our mutual information by subtracting what's called an average product correction, APC. And the average product correction is given by this equation. What is it? In the denominator here, it's simple. You have the average mutual information over the entire sequence over all positions, okay? The average. This is like a background value for the entire multiple sequence alignment, all residues. And then these are actually the averages corresponding to specific positions. Let's say corresponding to position I. If you consider its mutual information with respect to any column X, okay? So this value is just the mutual information between I averaged over all possible columns, all the remaining. If it's a protein of n residues, let's say, it is over an average over n minus 1 different columns. So that one, let's say this column I has its certain character, and it has a given mutual information with respect to everything else, and we want to get rid of this property. Same thing for column J. So we subtract these features that are not direct, that's like a background noise, if you like. So we have, by the way, I said summation, and I should have divided by n minus 1, okay? Because it's an average value. So I need to correct that. Any questions so far? All right. So I want to show a few nice applications to perhaps show you how things are easy, actually. And we can learn a lot biologically significant features based on this type of analysis. So that one is based on multiple sequence alignments of HIV-1 protease, a very important drug target. So at Stanford, there is a database. They have been accumulating the sequences from human HIV-1 protease from healthy patients, let's say healthy people, and also patients that have been subjected to different medical treatments. Okay? And we wanted to analyze them. So what we did is the following. First of all, the idea is you take the multiple sequence alignment and you generate the mutual information. This is like our heat map, right? We have residue index, and at each point we have our mutual information or the corrected mutual information. Or there are many other measures, but one of them to display what the correlations are. And then what a former student did was to take this type of mutual information which we can hardly see any signals, and it's a very easy thing to shuffle rows, columns, etc., cluster, if you like, groups, so as to observe this type of more distinctive patterns. So here the residues, they go from 1 to 300, I don't remember the size. But now here we have a different ordering of residues, like 1, 15, 28, with whichever cluster together, okay? So this is, and you can do that. There are many, many different methods to do this type of shuffling rows, columns, spectral clustering. We use one of them. Then what we did was to repeat the same analysis, actually, with untreated and treated sequences from the database. And these are the shuffled, reorganized. We generated the result for one of them, and then reordered the other. We generated the result for this one and reordered that one in the same way. And then we don't see the second cluster. Do you observe that? So what I'm saying, we have this common cluster, which is apparently common to either treated or untreated sequences. However, we have a new one now in the case of strains that have been, yes.
So what do you mean treated?
So these are
the change in gene expression?
Yes, there is this phenomena, when you subject a patient to a treatment, there is what's called MDR, is multi-drug resistance. So there is a mutation in HIV protease, and then which to acquire resistance, to drug. So when you have, the drug would be binding a certain region, and then the protein would be very smart to undergo a mutation to eliminate this effect. So the changes, we observe now a new set of co-evolving residues that are absent in treated patients. And we said these are most likely to be the MDR sites, multi-drug resistance. That's what is observed only if the patient has been subjected to a treatment. Okay? That's what happens where you have a resistance to drug. And I think this is already in itself, this is very valuable. You can identify now you can distinguish very clearly between healthy and sick patients, but this is essentially subjected to treatment or not any treatment for the same set of cohorts. Now, we have, where we observe, now when we look at the structure, the area where we have these type of drug-resistant mutations, compensating mutations, is right at the middle between the interface between the two monomers of the HIV protease. So HIV protease consists of two monomers, some of you may be familiar with, and we colored them with. So we have the drug-resistant mutations, co-evolving sites are at the center. And these that have been apparently mutating, changing in a correlated way, are more exposed to the exterior. And look at the mobility profile that we observe usually from the slowest mode. These regions will be naturally more constrained, and these will be more flexible. So that is, then we start to learn more. Actually, these are critical sites. And in fact, the drug would be binding at this region right below the flaps. And the mutations that would be acquired, if you like, after treatment are in this neighborhood at the regions that are consequential, if you like, at the regions that have low mobility, that are tightly packed. So if there is a mutation here happening, it's not going to affect the function of the drug. Interfacial regions are usually most important. So what we infer from that study is that we end up with two different types of correlated mutations, which we call phylogenetic and multi-drug resistant. They are located either exposed or interfacial regions. And the ones that have been selected throughout evolution without any consequence and retained, let's say, because they haven't altered the function, are essentially located at the mobile regions, highly flexible regions, as opposed to the multi-drug resistant sites that are highly restrained. Yes?
So I guess I'm wondering, is there a reason that certain patients are untreated and that also play into, like, is it that
of course, so I don't know the details how the data is being constructed, and this has been maintained for now quite decades at Stanford. But the idea is, I think, to understand better what is the genetic difference between, especially in response to treatment. What is the origin of multi-drug resistance? That was a big issue. Yeah.
But I guess I'm just wondering, like, could these mutations be correlated with something else, not the fact that
absolutely. And what I haven't presented here is that then we went one by one. Actually, we looked at each of them, and of the ensemble that I've shown here, a large majority, like 80%, have been reported to be multi-drug resistance indeed. I didn't present that part. If you go to this paper, you will see we have one by one all the compensating mutations comparisons, comparing with what's known from the literature, from previous studies, etc. Yeah, absolutely. Now let's go one step further. We have conserved residues. If you think of conserved residues, which residues would you think normally to be conserved in a given structure?
The weird ones.
Weird ones?
Like, cysteine, the ones that
Okay, let me ask in a different way. Which because they make some contacts, but of interest, what we are pursuing, trying to understand is whenever we have some allosteric effects, do we have co-evolving residues as well, which are not so easy to detect. And then we compare, the two sets of results, and we repeat for many enzymes. We wanted to extract a few patterns, actually, based on this analysis. And Evol is the software or the particular function under Prodi that is doing exactly this type of analysis. Suppose you have a query sequence, and you can extract the corresponding multiple sequence alignments. You can calculate the sequence entropy, conservation, coevolution, and you can compare with those based on structural dynamics. And in the applications, you will see many applications of it. Now, what is being observed is that we can plot, remember, we can plot the result from all modes, or the result from slow modes, or even the slowest, what we call the global modes. These are just shifted vertically. These are not absolute values. We just look at the profile. What matters is the profile. And here we have the profile of sequence entropy. And you can see already higher entropy means lower, more variability in amino acid type. You can see that the regions that tend to move more, these are the mean square fluctuations, also tend to have higher sequence entropy. Okay, that's only for one example, actually, we, for fun, we did in the same way as we plot this color. We have those color-coded structural dynamics. And this is very easy by replacing the column for B factors in the PDB file by those values, the mean square fluctuation values. We did the same thing. We use now the sequence entropy to color code our structure. And you can see how actually higher entropy goes with a higher mobility. Okay? Yes?
I know that really portions in the core are expected to have less movement.
Yes.
This for similar reasons, those residues in the core are expected to have less variety than the rest. Is it possible to generate that, trying to remove this effect?
That's a very good point. We haven't done that, but I think it's an extremely nice point. So try to see what remains, or where the differences. We can have some kind of a normalized plot for one and for the other, and try to see where the differences are. That's, of course, you're coming from that perspective. I have biased you. You're thinking everything is dependent on the mobility, but where are the differences? We were not. In the beginning, we were not thinking like that. On the contrary, we were saying, are we going to observe any correlation? And this is one of the good examples. You can't see that in all applications. But I will show you also the result from all the application to the entire data set. We have this trend for sure. So I think that's a good point to further elaborate. We didn't do that. You can do it. All right. So, and this is the result from all now residues, 8,000 residues in our data set. And we were plotting the information entropy versus the effective mobility. And these are the error bars. So what we did, if you look at the individual values, they are quite scattered. But we took grids of that size and plotted the average values in each grid. So you can see that the way, what do we see? The entropy definitely increases with mobility, but the increase in entropy starts after a certain threshold almost. It's almost flat here. So that curve, the red one, is based on the global mobility, and this one, where the effect is weaker, but we have the same effect, is based on this is the mean square fluctuations, all modes, all the n-1 modes in GNM. So, maybe you also noticed whenever there are some signals, it is, and if we start to dissect the different modes, if we try to see what is happening in the global modes, always the signals are magnified. When you start adding all modes, things start to average out. The effects are weaker, but still we can see pretty well the trend. Okay. And that was the distribution of residues. You know, how many residues did we have in different grids? Of course, the error bars are large here, as you can see, and this is simply because we have very few residues in that interval. But essentially, that's the pattern. So another thing that we were considering was we focused then on hinge sites, right? And in this structure, it's very easy to observe, to see. So this structure comes like that. So there's one domain, and then it's connected here through this residue to another subdomain, another helical hairpin, if you like. And this, when you do the calculations, the ENM calculations, you just observe this to be the hinge site. And there is a very clear-cut way of doing it. When you plot the eigenvector itself in GNM, you go from, let's say, positive region to negative region. There it crosses the zero line. So it's very unambiguously defined where the passage from one direction of motion to another takes place. So that is the hinge side, and here. So these are residues that we found actually to be highly conserved, and there are many. These are just examples. To be highly conserved and also playing a key role. And we were very interested. You were matching the type of residues that cysteines might be more conserved. Here we have observed that glycines, they pop up quite often. And they have to enable this type of, you know, because of their extra rotational ability, they're often located at hinge sites. Or, for example, a proline may be important because it forms a kink exactly, it changes the direction, etc. And another issue was the referee told us, oh, all your conserved residues are most likely to be buried, and what you are observing is essentially if you have something that's very buried, it's going to be more conserved, etc. And the reason we chose these in particular, I think we included in the appendix, so I don't remember, where these are just cases. You can't say that these residues are buried. On the contrary, they are exposed, quite exposed to the environment. It's not quite intuitive unless you start to, you know, really analyze the collective dynamics, etc. And we colored by different colors the two portions of the chains, or three portions separated. So, for example, if we look at this structure, this green portion is then connected to the violet through this P73. So this is like the interface between different substructures, etc. Okay. And another observation that we made, and this is, again, something to be further investigated, at least we have now some patterns that need to be further established, is that when we do the coevolutionary analysis, the regions where we had strong signals, strong co-evolving amino acids, in many cases, first of all, people have been using these things for helping with structure prediction, you know? Because if there is a pair of residue that happens to be co-evolving, shows a very strong tendency, intuitively you would expect them to be making tertiary contacts in the 3D structure. And of course, you all know that if you have a sufficiently large number of constraints, you can predict the structure. This is the way NMR models are being constructed, for example. So if from those analyses, suppose we have now a series of very strong signals telling us about potential contacts, and if you use this as input in your folding simulations as bias, that could be helping the... So, and this is what I'm talking is not a fiction, that's a reality. Now, there are very nice studies that have been recently published on direct, what's called direct correlation, where they remove all sorts of indirect effects and noise. And they are identifying what are the strongest signals that are potentially making contacts, especially for membrane proteins. And this, using this type of constraints, now it helps a lot in the predicting membrane proteins, which is always a challenge, as you know. So that is the state of the art. If you can take a look at these things, how they are being used. Now, of course, one can go one step further. Now it becomes more challenging if you have a pair of interacting molecules. Okay, if you have a pair of interacting molecules, and if somehow you generate the multiple sequence alignment for protein A and B, always belonging to the same species, okay, you can do that. And then you can start to observe perhaps co-evolving positions. What it's going to tell you, the co-evolving pairs then. Then you have information on intermolecular contacts, right? Some, at least some possible, probable intermolecular contacts. And that is even more important to understand who is interacting where. So here, what we observed in this case, in this protein, was this, wherever we had this strong signal, turned out actually to be where this particular enzyme recognized DNA. So we had a bunch of many co-evolving residues at that surface, which is essentially the DNA recognition site. Apparently, facilitating in a correlated way the recognition. And here's another example with cathepsin B. Again, we have a very strong signal here. And when we plot the mean square fluctuation, or the mode 2 in this case, we have at the same region where we have a very strong signal, we have a high mobility. So this is the protein, and in cyan is its substrate. And in red is the region where we have the strong signal. So again, this high mobility and its sequence variability we propose to be required for binding the substrate, for substrate specificity. Okay? So that concludes my section. The major point I want to make at the end of all this discussion is you can have many different types of functional sites: chemical, core, hinge size, or recognition, substrate recognition. And in the case of catalytic sites, the mobility is minimal. It's as if they are not really moving. They are just very precisely positioned to enable the chemical reaction. They need to be very precise. Very high fidelity is important in catalysis. And what's happening is at another level. It's not like a collective motion, but a local, more quantum chemical events that become important for chemical reactivity. Now, in the case of a hydrophobic core, again, we have minimal movements. They're conserved, and they confer high stability. Hinge sites, they don't move, again, as I explained yesterday. But they are conserved in many cases, and they confer rotational flexibility, not translational, rotational flexibility. And the substrate recognition, and I'm talking now about transient complexes, substrates that not part of a multimer. So they have usually high mobility and high coevolution propensity, and that's required for their adaptability. Okay, these things are the most important things I wanted to emphasize. Now, this ends my lecture before we move to the next room. However, I thought there were a few questions yesterday. Some of you asked me about the allosteric communication, etc. And I prepared 5 slides for that, thinking that this might be of broader interest since many questions came up already. So this is what, and because it also ties very nicely to what we've seen yesterday. So allosteric communication mechanism. Whatever I'm explaining, whatever I'm starting to share with you, I'm trying to share with you, are very, very simple things, to implement, to program. And everything is actually, because it is, you build the theory, and once you have the theory to implement and to do calculations, applications is extremely easy, and why not to use them, as you will see. So the idea is, now that we have a network model, And suppose we are interested in signal transduction, transmission. Suppose there's a substrate binding here in the extracellular region, and then you have a signal in GPCRs happening in the intracellular region. So you want to see how the signal propagates, let's say, from one point to another, given a network model. Okay, we've been doing everything with it. So the assumption here is to use a Markov process. So it is, a Markov process will be stepwise evolving from one to another. So in space, actually, what's happening is through a sequence of adjacent contacts, it propagates. And we still stick to the same definition. We define the connectivity by the Kirchhoff matrix, and we have them as a... In other disciplines, they call the same matrix, they call it Laplacian, etc. And you can see the theory here. Now, what we are doing is very, very simple. And I hope you will agree. So this is the connectivity matrix. Remember, the connectivity, the Kirchhoff matrix, the way we defined, we put -1 to all off-diagonal elements whenever there is an interaction. Now, instead of -1, I could call it an affinity, a certain, because it's a measure of the strength of interaction, like a force constant, right? We call it an affinity. And you can, you don't need to use 1, or you can use any force function parameter. And we just call it affinity matrix, and because this is the terminology in computer science. And the diagonal, remember, one of you said yesterday, this is the degree, right? We call it the degree diagonal matrix. So our Kirchhoff matrix, we are just writing as a difference between the degree matrix, diagonal matrix, and affinity matrix. So it's the same thing. Our single matrix, we now split it into 2 parts. Very simple. Well, the thing is, this A, for those of you who are familiar with Markov processes, you know perhaps that when a master equation formalism, is there anybody familiar with master equation formalism? No, okay. All right, that's fine. So we, in a Markov process, everything, in the same way as the Kirchhoff matrix defines our dynamics, in a Markov process, everything is defined by the conditional probability of signaling. So given that you have an effect here, what's the probability of observing the effect at another site? This is the transition or conditional probability. So this transition probability, conditional probability of signal transduction from residue j to I, we designate as Mij. And it is simply defined in the network model in many other disciplines as the affinity divided by the degree. So remember, the degree is the summation over affinities. Okay? So I'm a central node. I'm interacting with many nodes. I'm node J. Okay? I have many nodes, and my interaction with each of them is Aij. And the summation over all interactions is Dj, so strength of interaction, probability of interaction. This ratio would be, would just give me the probabilistic occurrence of interaction with I, simply, among all possible neighbors. Are you with me? Yes?
In this model, though, aren't all Aij's like ones? No.
Mine's one, yeah. Yeah, you can take it as 1.
So all Mij's would be basically the
So if you take like the original simplest Kirchhoff matrix, all the Aij's will be -1, yeah, and Dj will be the summation of over them. So this is like you are assigned the probability of interacting with one of the neighbors is 1/5 if you have 5 neighbors, simply. But if you have different grades of interaction strength, you can incorporate them in. So that's all. As simple as that. Or you can write it is in a matrix form, where you have the affinity matrix composed of that, times the inverse of the diagonal matrix, which is also a diagonal matrix, obviously. So that is this transition, Markov transition matrix fully defines what's happening. If you have, let me see. So here, if you look at this equation, the probability of occurrence of a signal at a given point, provided that you perturb one of the residues, is simply given by this function, which is also time dependent. So I will leave it there just to show that it's very simple. But also I will go further a little bit for practical purposes what we do. A metric that's being used, and again very established in other disciplines, is hitting time. Suppose you take this simplest network, okay? IJK. It can't get simpler than that. A network of 3 nodes and 2 springs, okay? And suppose I would like to assign for each pair of residues, I want to assign a certain value, a certain metric that's going to give me the probability of signal transduction, okay? So it's called hidden time, but it's not time actually. It is just, but it's proportional, if you like, to total number of steps that needs to be undertaken to go from one residue to another. So in this case, this simple example, let's take the hitting time from J to I, okay? Suppose I perturb J, I bind something here. How long does it take for I to sense it, okay? Now, the way to calculate, there is a direct path, but there may be many, many indirect paths in your network, right? So actually, you can consider this path as one path from J to I. This is one of the paths, and it consists of one step. And if you think of equal probability for each steps, its probability is 0.5, a priori probability, okay? But you can have also, you can go from J to K and back to J and then to I. This is possible. Or you can go from J to K, back to J, and then again to K, and then again to J, and to I. And many of them like that. So all possible combinations. That's, in theory, that's possible. And each of them has a different number of steps and a different path probability. Suppose you go from J to K, the probability is 0.5. From K to J, there is no other alternative. The probability is 1. And J to I is again 0.5, so the overall probability is 0.5 squared, and so on and so forth. So the hitting time is a summation over the length of all such paths multiplied by their a priori probabilities. This is this metric. So you have one step with this probability. You have another path of 3 steps with this probability, etc. You have a summation. And it turns out that this summation is simply 3. Very simple. Those who are mathematicians may see it easily. So that is the hitting time metric in this simple network to go from J to I. Now let's go from I to K, okay? Let's go, yes.
Yeah. I don't quite understand what you mean by perturbation, because if you perturb one,
yeah,
so that would be the J.
Suppose,
going
yeah.
from J to K is not perturbing K?
Sure. So what I'm interested in is, suppose I have some kind of perturbation at a given node. How fast the other node sense it. So suppose this is my binding site here at a given residue. Which other residues will be most efficiently affected by it? There will be a perturbation in their positions. Okay? So that is to identify allosteric communication paths or mechanisms. Yes.
I don't know if you've done that.
Maybe not. She's
Well, it's okay.
okay.
So I guess what I'm wondering is why you have to why J can only affect I or K and not both
Well, this is
simultaneously.
and not affect what?
Why it can't affect I and K simultaneously?
It can affect I and K. What we are trying to calculate here is a metric for affecting I starting from j. This is HJI that we are pursuing. This is what we are calculating. So for each pair, we're going to have a hitting time, okay? And there is a starting and ending point in each case, okay? And you can do that also for this one. I'm not going to go. And in this case, it turns out to be 4. Now, you can't do that with a real network, right? It's very time consuming. But you can do it very easily, actually, using R. And I'm not going to go that. So this is the generalization of the formula, but I'm not going to go into that. Or another generalization. You can write this formula, actually, and there is the whole derivation in this paper. You can write it in terms of the inverse of the Kirchhoff matrix itself, very easily. And what is also interestingly, this is path dependent. So you can, for example, when you start from I to j, you have a certain speed, if you like, for signal transmission. But the opposite is not true. If something has many neighbors, your initial node has many neighbors, it's going to propagate the signal everywhere, as opposed to something that has fewer neighbors, let's say. But in some cases, we want also to have a matrix, like a heat matrix, which is symmetric. What we do is we sum up the HJI and HIJ. So you go and you come back, OK? So what is the commute time, it's called? The commute time is simply given by this equation. Now, what I'm trying to say is that suppose you have an even system, even a very complicated one. Building the Kirchhoff matrix is an instant thing, immediately. Knowing the Kirchhoff matrix, you can immediately calculate what are the commute times between all pairs. You have a heat map, and you would know which pairs of amino acids are most efficiently communicating in that network. Are you with me? I think that's very beautiful and useful, simple. And if you look at that expression, remember from yesterday, gamma II minus 1. This is a question now. What is the physical meaning of it? The diagonal term of the inverse Kirchhoff matrix is a measure of? Yes. Yes, the mean square fluctuation is the diagonal element of the covariance. So this is nothing else than delta Ri square. This is delta Rj square minus 2 delta Ri dot delta Rj. It's proportional to it. So delta Ri square, delta Rj square minus 2 delta Ri delta Rj is nothing else than the change in the distance fluctuation, delta Rij square. Okay? So there is a lengthy derivation in all that. But the commute time, essentially, is nothing else than the fluctuation in the distance between two residue. It's just proportional to it, I would say, there is, if you forget. So that ends my presentation. Oh, there are applications, and this is really beautiful. You know, if you look at this commute time that we plotted, and what we don't want to deal with a two-dimensional map. We projected it, everything here, to a one-dimensional, just taking averages of columns. And for that case, this is a phospholipase A2. We know the catalytic triad residues. And we wanted to see where they are located. Not surprisingly, catalytic residues have a minimum average hitting times. So they are located at such positions that they're really central to the structure. You see what I mean? So this way you can identify what are really the central residues that mediate, that control almost the entire structure. So that was a good application. I'm not going to, and the one can, this was also, we were comparing the physical distances versus commute distances. And you can see that there is a really broad variation. Physical distance doesn't necessarily mean fast commute distance. It depends on the network architecture. And there are many cases where, you know, these neighbors have the same distance, whereas they have very different, the two yellow dots, they have very different commute times. Or vice versa, you can have cases where you have very, you know, the same commute distance while the distance on the structure is quite different. So such cases. And if you plot the hitting time, you know, the average versus the standard deviation for all residues in a given structure, and we've done for a series of structures, you can see that the catalytic residues are always are really located. They are very efficient. Small hitting time is very efficient. And they are also, they don't vary across the different residues. They are very precise. We call it efficient and precise. That ends my presentation practically. I will say a few concluding remarks. What we think is that proteins are designed to favor functional changes in their structure. I'm not advertising intelligent design, but I think in this room I can say safely that they are really designed to favor functional events. So pre-existing soft modes would be facilitating their substrate binding, their activities. And we know that the collective dynamics is very important for function, for allostery. We have a high flexibility at the recognition sites. By the way, this is the GROYEL that I've shown yesterday in different conformations. This is an artistic rendering from a previous PhD student of mine, which also became the cover of PLOS Computational Biology. He was inspired by the posters of Kandinsky that we had in the lab. You didn't have the opportunity to come to our lab this time, but perhaps in another occasion. So the correlated mutations at recognition site enable substrate specificity while conferring conformational adaptability. And of course, there is no need to say that for target druggability, it's important. My last message, something that I've been showing for 20 years, is what I fully believe is there is a very nice balance between mechanics and chemistry. So many molecular entities are mechanochemical entities. And so when you deal with small size systems like drugs, chemistry matters. Chemistry is really important. But as you go to larger and larger systems, and we could have here, for example, the viral capsids even larger, gradually you have in those systems, you will have increasingly a dominance of molecular machinery, okay? And as you go in this direction, you have chemistry. So in modeling, I think the most important thing is to really strike the right balance. You don't want to put too many chemical details if really what matters is the molecular machinery or the collective dynamics. And in this case, you can adopt coarse-grained approaches. So it depends on what you're observing, the time scale, the size of the system, etc. And I'm going to tell a few bad things now before finishing. I think it's like an art to be able to use the right tool for the right problem. For example, if you are interested in full atomic interactions, and MD is very powerful. If you want to go, and even for very large systems now, you've seen how you can do simulations. If you want to go to really long time processes, well, you need some coarse graining. A&M is one of the possible approaches, which we found very useful. Now, that was also one question, I think. Not all evolutionary correlated sites are necessarily physically meaningful, we've observed. They don't refer to any structural correlation or dynamic correlation, although we obtain lots of correlated sites when we do this coevolutionary analysis. We have lots of noise. Our role is to clean, this type of false positives. Of course, what Ahmed presented was beautiful, and we can use for computer-aided drug discovery. But a real quantitative evaluation of binding affinity is still a challenge. So we know which regions have higher preference, and based on that, based on the ensemble of probes that tend to cluster in a given region, you can build pharmacophore models, you can estimate binding affinities. But we're far from having a well-established method for quantitative evaluation. again, someone asked, what about using the real drug instead of the probes? So we essentially, we're very confident that we can identify druggable sites. But when it comes to the real drug that needs to be attached there, this is another, again, you may build some pharmacophoric models. You can, using your probe composition, you can try to design something. However, this is it's all about identifying whether a protein is druggable and what are its druggable sites. Last and not least, I would like to acknowledge many people. Have you been to the point, by the way? No? Yeah, that's something we have to also change. You need at least one free afternoon, right? Of course, the weather I can't change. The weather is not cooperating. But the point is gorgeous. You should. So this is, somebody was asking me, this is the center of the city. Here. This is the Allegheny River and Monongahela River. And they merge to form which river?
Ohio.
And the Ohio River goes all the way to, merges with Mississippi. So this is all about geography. So it is, we are somewhere here, over there. And I live somewhere here. On the north, this is the north, this is the south, and this is the middle. I'm in the North Hills. So this, and I would like to acknowledge, of course, in Ahmet and Tim and Anindita. Eran was a former postdoc who really did a lot in building methodology. He's now heading a bioinformatics institute in Tel Aviv, Tel Shiva Medical Center. And these are the folks with whom we actually started how many years ago? I can't even calculate. We built the elastic network model methodology more than 15 years ago. So I will stop here and to let you ask questions and also let you do hands-on experiments. Yes?
Thanks for that great lecture. I was wondering when you presented the new conditional Markov matrix
Yes.
building, right? The basis of it is already the information-bearing network from the evolution correlated network?
No, the basis of it, everything is around this, what I call the gamma matrix. What I introduced yesterday, the Kirchhoff matrix, connectivity matrix. Okay, and then this matrix you can generate very easily.
It's already incorporating the physical constraint of
Yes, the entire topology of the network. Yes, yes. So because the gamma matrix, Kirchhoff matrix, is a measure of the. You said that it looks like a contact map, right? So we are using this matrix that looks like a contact map. And this matrix, it's so easy to generate it. And using it, you can calculate immediately the commute times. And yes?
So do you find pathways of, so in the commute time heat maps, for example, do you find pathways that you can trace through those maps that give you some idea of, let's say, real allosteric pathways?
Absolutely, absolutely, absolutely.
So
yesterday
low commute times, or is there some characteristic commute time that
they are. So we are trying to identify usually the most efficient, smallest heating time or commute time pathways. Yesterday I was mentioning to you this salt bridge at the interface, which is very important. Actually, we identified the salt bridge based on this type of analysis. That's what we applied there. So we know there's an ATP binding site here in the lower ring, okay, in the subunit. And then there's an intermediate ring of 6 subunits, and there's a cap. Now you want the ATP binding to be sensed by the cap all the way, I don't know how many angstrom away. And we were thinking, okay, these are residues that bind the cap, and this is the ATP binding site. I have now those 2 endpoints. And we tried to identify what would be the shortest path based on this type of commute distances. And it was going exactly through this salt bridge, critical salt bridge. Now, this is when you perturb the salt bridge in the mutant, and now there is a redistribution of interactions. You're now interfering with this effective mechanism of signal transduction, and experimentally it's observed that the allosteric cycle is being arrested in that particular position. So that is, I think, it's extremely useful, I think, when you start to focus on a given system, or even if you want to do things serially. This is so fast. Why not to do it, as an extra piece of information in addition to other things that you do?
Well, Ramanathan has been trying to identify
Absolutely.
these paths,
Ska.
but using coevolutionary patterns rather than the kind of, like, I wonder if you
I know.
to coevolutionary latency.
So what I know what his method, statistical coupling analysis, is. Ska. It's a very nice method, although there are now better methods. I was mentioning this direct correlation. It's essentially he's looking at co-evolving amino acids, but I don't agree, and I think there's a very serious point to make here. So his basic assumption, his postulate, is that the signal will be transduced only through co-evolving residues. To me, that is the starting assumption, right, in there, because they base it. That's wrong. On the contrary, it's going to involve many conserved residues. And conserved residues, they don't appear as signals in co-evolving, co-evolution maps. So I think there is a big issue one needs to take into consideration. It's not enough to base everything on co-evolution. So if you think of the most important property, the first thing you need to think is conservation. And then coevolution is like the next thing. So this is not only my opinion, by the way. It is this has been criticized because of this. Okay.
So when we have a protein in a particular state, let's say nothing is bounded, it's not a single conformation, but an ensemble of
That's right.
conformations, right? And then we have a different state in which it is, let's say, bound to a ligand, and then it has a different
Ensemble.
of conformation that it adopts.
It's biased
So,
now. There's a population shift, if you like.
Yes,
Yeah.
exactly. Can we get information about this from a normal mode analysis point of view, looking at that? Or if you compare the two systems, would this analysis tell us something about the different ensemble conformation, that there is a difference between them? One, and then how does sequence variability, how does it relate to this change in ensemble conformation? Like, for example, can we say that you mutate one residue and it may not change this ensemble conformation is
That's right.
characteristic for that particular state, but then mutating something else may
That's right.
change it, so.
Yeah. These are very, very good thoughts, by the way. And of course, I'm going to give my answers, but these require also, each of them requires some critical studies. Now, with regard to ensemble of bound and unbound conformers, they are, in terms of the conformations that are being sampled, they would be more or less the same, but the population changes. So something that's, let's say, a closed form, which is not perhaps very probable in the unbound form, will be stabilized and have higher probability in the bound state. Okay. Now, how the normal modes can help in that? Well, we have shown in many studies, and many other groups as well, that this mechanism of opening, closing is nothing else than a movement along mode 1 or mode 2. 90% of cases. So if we know that this is, we have an open state, which is the most probable state in the unbound form, we will know exactly the way it tends to reconfigure. And one point we are trying to make is not necessarily all the alternative conformers coexist instantaneously. But what is possible is this ability to change its conformation. So if originally you have the unbound conformer and you have a substrate that comes, even if it were not predisposed, ready, it will have this disposition to close down. So that is, then this will be, and this type of predisposition to undergo certain conformational changes is exactly what elastic network model is predicting.
So if you just look at the normal analysis of the bound and unbound state, you would see a difference in the predisposition of this?
No, if we look at the bound and unbound state, and yesterday I showed an example, and suppose now you look at the deformation and change in conformation. You superimpose them and you look at what is the deformation, okay? What's the change in the coordinates. Now you see that as, you have this 3-dimensional deformation vector. What we are doing, knowing those two endpoints, if we know them, we compare them with our modes, with the eigenvectors. And we have seen that they agree with one of the top-ranking softest modes. Now, if you would like to make a prediction, suppose you don't know it, then, there is, when we start with the unbound, with the less tightly packed conformation, it has higher mobility and it's easier to make predictions. We start with the unbound form. We do our analysis. Now we generate the eigenvectors 1, 2, 3. And now we deform our structure along this eigenvector. But there will be, of course, we don't know exactly the size. And we don't know also which eigenvector. There may be an ensemble. What we can do is to generate an ensemble of probable conformational changes that might be stabilized in the bound form. Then you can use this ensemble, and that's a very common thing people are doing, to have a set of alternative conformers in your docking simulations, and try to identify which one among those probable alternative conformers fits your ligand. Okay. I think we are ready at this time to go to the other room. And again, whenever you would like to take a lunch break to take a walk, feel free to do whatever you like. But we will try to, make best use of the remaining little time. Okay? Thank you very much.